This tutorial will take you on a brief tour of the structure-function linkage database, which links enzyme sequence and structural information to specific functional information. I'll start with a brief introduction to the SFLD using the radical SAM superfamily, a diverse group of evolutionarily related enzymes that use a conserved 3-cysteine motif to bind an iron-sulfur cluster and activate S-adenosylmethionine, as an example. Then I'll show you how you might use the database to make some hypotheses about the function of an uncharacterized enzyme. There are a couple of ways to get into the database. I'm going to start by browsing through the available superfamilies. Sequences in each SFLD superfamily are organized hierarchically based on sequence, structure, and function. At the top level of the hierarchy is an enzyme superfamily. The next level of the hierarchy is a subgroup. In the radical SAM superfamily, for example, we have 52 different subgroups, which have been defined based on overall sequence similarity, so that enzymes in each subgroup are more similar to each other than they are to other enzymes in the superfamily. The next level down in the hierarchy is family, where again, enzymes in a family have more sequence similarity to each other than to other sequences within the parent subgroup, but more importantly, they're thought to catalyze the same overall reaction based on the same mechanism. We currently have 74 families defined for radical SAM. Now I'm going to click on the link to take you to the radical SAM superfamily page. At the top of the superfamily view, we have some basic summary information about the superfamily. You can see the total number of non-redundant sequences in the superfamily. These are subdivided into those with a family classification, in other words, those where we think we know the function, and those without a family classification, in other words, those where we don't know the function. You can also see the total number of structures and reactions. On the left, an example superfamily structure. Just under that, we have a brief summary of the function of the superfamily. The References tab gives you a few literature references for further reading. These aren't meant to be comprehensive, just a place to get started. The Partial Reaction tab shows you the chemical capability shared by radical SAM superfamily members. And the Cofactors tab shows you the cofactors used by all enzymes in the superfamily. In this middle section, you can access the curated alignment for the group and also some download tools, but I'm going to show that to you a little later. Skipping down to the bottom, we have a tabular view of the way the sequences and structures are distributed between subgroups and families. Here, subgroups are shown in blue, and families are shown in yellow under the appropriate subgroup. The column with the T heading lists the total number of sequences in a given group. The column with the K heading lists the number of sequences in a group that have family assignments, in other words, those where we know the function. The column with the U heading lists the number of sequences in a group that have not been assigned to a family, in other words, those where we don't know the function. And the column with the S heading lists the number of X-ray structures with at least 95% identity to a sequence in the group. All of these names, subgroup names, family names, reaction names, and sequence and structure counts are clickable links. So if you want more information about anything, you can click on the associated link. That's a very brief introduction to the SFLD, and now I'm going to show you in more detail how you might use it. For this scenario, I'll start with an enzyme that has no associated experimental information and show you how you could use the SFLD and Cytoscape to make some hypotheses about what its function might be. This is the NCBI page for an enzyme annotated in GenPept as an L-lysine 2,3 aminomutase. But if you look at the references section, these papers are all large-scale sequencing papers, so the annotation doesn't seem to be based on experimental data. I'm going to get the protein sequence for this enzyme and copy it and then paste it into the SFLD sequence search box. The sequence is then searched against the hidden Markov models in the SFLD. Hidden Markov models are models based on sequence alignments, which give you the probability of finding a particular residue in a particular column of the multiple alignment. The SFLD hidden Markov models are based on our curated multiple alignments for all the families, subgroups, and superfamilies in the database. What we get back from these search results is a list of the groups that the sequence matches, with the best matches listed first. For each match, I have the name, what level of the hierarchy the group is from, in other words, whether it's a family, a subgroup, or a superfamily, and the associated e-value and score. Our top hit is the glutamate 2,3 aminomutase family. The L-lysine 2,3 aminomutase family, which the sequence was annotated to in the NCBI, is actually the third hit. The second hit is the PLP-dependent subgroup, which both glutamate 2,3 aminomutase and lysine 2,3 aminomutase are part of. So just based on score, it looks like the sequence is more similar to the glutamate 2,3 aminomutase family. But let's look in a little more detail at how well the sequence fits into the two top family hits. I'm going to start by clicking on the link to examine this sequence in the context of an alignment of the lysine 2,3 aminomutase family, because that's what it was annotated as in the NCBI databases. So here's the curated alignment for that group, and the sequence of interest has been added in at the bottom. The catalytic residues required for family-specific functionality are listed in this table at the bottom of the page. Each residue is associated with an evidence code, which tells you what kind of information was used to determine its functional role. Clicking on any of the evidence codes takes you to a page in the SFLD that gives the definition of the evidence code. And when applicable, residues are also associated with a literature reference. The catalytic residues are also highlighted in the multiple alignment, so it's very easy to scroll through and see if our sequence of interest contains the residues known to be required for family-specific functionality. And although this sequence does have most of the family-specific catalytic residues, in the place of these two aspartic acids required to bind the lysine substrate, it has a lysine and an asparagine. So based on this alignment, it looks like this sequence is probably not a lysine aminomutase. Let's see if it fits any better into the glutamate 2,3 aminomutase family. I'm going to return to our HMM search results and then examine the alignment of our sequence with the glutamate 2,3 aminomutase family. Again, the residues required for family-specific functionality are highlighted in the alignment. Scrolling through, we can see that in addition to good overall sequence similarity, this time the residue does indeed have all the residues required for family-specific functionality, including this lysine and asparagine required to bind the glutamate substrate. So it does look like this sequence is a glutamate 2,3 aminomutase. So let's get some more information about this family. This is the family page for the glutamate 2,3 aminomutase family. The family doesn't have a solved crystal structure, so there's no image here, but there is summary information and an associated literature reference. At the bottom of the page, you'll see the reaction catalyzed by all sequences in the family. If you want to look in more detail at the sequences in the family, you can click on the sequence link at the top of the page. That will take you to a table listing all the sequences in the family, along with some associated annotation information like species and links to various other databases. You can control what information you see in this table using this Toggle Columns tab. Some things you might be particularly interested in are operon context information from Microbes Online, length of the enzyme, protein data bank identifiers, etc. You can download this information as a tab-delimited file, which can be opened in any standard spreadsheet program like Microsoft Excel, or you can download the sequence set. This concludes our brief tutorial on the SFLD. For information on how you might use the SFLD along with the Cytoscape Network Visualization Program to visualize sequence relationships in large groups of enzymes, check out Part 2 of this tutorial series.